We were able to, I mean Richard Deans was a chemistry student in the lab, together with Chetan Khosla, who synthesized this compound, and we used the fact that it's toxic to actually find its target. So we could add the drug. We knew that it caused some growth phenotype, and we could look for modifiers of it. And the reason I'm spending some time describing this setup to you is because what we did was to actually systematically compare an shRNA screen with a CRISPR screen, and it was actually very interesting to do this comparison. So, the screen workflow is exactly the same as what I described to you. In both cases, you're infecting a lentiviral library. In this case, one shRNA per cell. In this case, one guide RNA per cell. And we do parallel screens. One pool gets treated with the drug, one pool is untreated. At the end, you do sequencing and you compare and you ask which genes, when knocked down or knocked out, modify the activity of this drug. To describe what is really, I think, the most interesting part of this story. So what I'm showing you in this plot here is the phenotype of the shRNA screen on this axis and the CRISPR screen on this axis. And so to make a very long story short, the target of this drug is this gene, this DHODH, is a critical component of de novo pyrimidine nucleotide biosynthesis. So viruses, when they infect a cell, massively upregulate nucleotide synthesis so they can support their replication. If you can't do that, then the virus can't replicate. And so that's why this was actually an effective antiviral. Unfortunately, it's also toxic because cells need those nucleotides to support their own DNA replication. It turns out there's actually quite a nice therapeutic window there where viruses need a lot more nucleotides than a standard cell does to stay alive. But the interesting thing here is that you can see when you knock down this gene with an shRNA, you could actually, it's one of the strongest sensitizing factors to GSK983. And that's how we were able to find it, is because we could knock it down and show that it actually sensitized against the drug. In a CRISPR knockout screen, this is an absolutely essential gene. So if you delete it, those cells just die. So really there was no phenotype in the CRISPR screen because by the time we actually took that measurement, several weeks had passed and all those cells, for the most part, had dropped out. I think there's actually some interesting counterpoints to this idea. I think a number of people have now seen in a CRISPR screen, you can sometimes see a modification of an essential gene by getting, in some cases, hypomorphs that give you, deletion of one allele or two alleles in a cell line that has 5 alleles. So there are some counterexamples, but I think we've seen in a number of cases this very interesting difference between, the utility of a knockdown phenotype, which you might get with an shRNA screen or a CRISPR-I screen, and a complete knockout where you're really eliminating all of the function of a gene. Now, conversely, these genes, which are negative regulators of mTOR, only showed up in a CRISPR screen. So the best shRNA in the world might give you 98% knockdown, and if that is not enough to give you a phenotype, you won't see it. So these two negative regulators of mTOR, which ironically actually show up in just about every screen we do for some reason. mTOR is a very important gene that's involved in a lot of different processes. Only could be found with the CRISPR screen, so you really need complete deletion. So the interesting kind of take home from this is that sometimes it's good to have knockdown and sometimes it's good to have knockout. And we've actually found by comparing the two screens, sometimes you get a much more complete picture of the biology. Just as a side note, we found not only the de novo synthesis pathway, but the salvage pathway. So cells have two ways of taking up nucleotides. They can either make them themselves or they can take it up from the outside. We found both of these pathways in the screen, and we're now exploring a combination strategy where we block de novo synthesis and salvage to actually improve the activity of these compounds as antivirals. One other side note, by way of comparing shRNA or knockdown and CRISPR knockout screens, is that we've actually, when we've done these types of comparisons with the same libraries side by side, it's not that one library is crap and the other one is good. They actually both do a pretty good job of calling essential genes. So if you take a gold standard essential gene set, so these are genes that have been found in like 100 cell lines to always be required for growth when you knock them down, in this case with an shRNA library, and you say, that's my gold standard, and so whenever I find that, I'm right. If I don't find it, I'm wrong. And you draw this type of ROC curve, so up and to the left is better. You can see that they actually, both the CRISPR library and the shRNA library actually do a pretty good job of calling essential genes, but when you combine the two, you actually can do better, which means that there's non-redundant information in those two screens, and it's actually useful to combine the two. Yeah?
So, designing basically for particular crappy guide RNAs that are more likely to cause like a -3, -6, -9. So more likely to give you that intermediate knockdown phenotype. So you make a slightly crappier protein, but you don't get as much output.
That's a really good question. So in some cases, I think if you're targeting the protein coding region, it's not a trivial problem to figure out where exactly you would put those guide RNAs to have a crappy effect. I'll show you a study where they actually scanned across a couple protein coding regions to find what would be good. And maybe in those examples, you could say, well, this would probably be bad or relatively bad so that you'd have an array of phenotypes. I think in CRISPR-I, it may be easier to conceive of a system where you would really try to, I don't know how you guys would think of doing this, but stepwise move away from the promoter until you realize that, instead of having, as efficient knockdown as you can, you have, a range of expression levels. There it might be a little bit more uniform to do that kind of thing, but it's actually an important question because sometimes you really would like to have a range of phenotypes. Yeah. By the way, feel free to interrupt. I realize I'm kind of rambling on here, so if any of you guys have any questions, feel free to shoot away. Okay, so one of the nice properties of this system for drug target identification is you don't need to modify the drug. So, typically if you want to find the target of a drug, you'd have to stick a crosslinker on there. You potentially destroy its activity. It's not sensitive to the strength of the interaction, and you can find those targets in the biological context. Okay. So in the examples I've discussed so far, typically the guide RNAs have been targeted to a protein coding region. But there have been a number of other screens that have been done where people have started to look at the non-coding genome. So, of course, in the context of a CRISPR-I, you're typically targeting upstream of the transcription start site. But in a number of studies, people have now used active Cas9 and actually increasingly a number of other of these variants that Luke described to really start to probe what are the functions of not just protein coding genes, but actually regulatory elements. And so this is a study that was done in Feng Zhang's lab, for example, where they tiled 100 kb upstream and downstream of this gene, which turned out to be one of these genes that they had found in their previous BRAF screen. And the workflow is effectively the same. You take this library of guide RNAs, now not targeting the protein coding region, but targeting a very large region of upstream and downstream regulatory elements that control the transcription of that gene, and ask for which of those elements might actually affect the transcription of a gene. Earlier on, actually, this group and Dan Bauer's group, together with Stuart Orkin, did a quite similar screen. I happen to like this diagram better this way, but these guys actually did it first, to tile, again, the regulatory regions that are around BCL11. So this is a gene which has an enhancer that actually affects the transcription of again, one of these fetal hemoglobin genes. So in this case, they're looking for modifiers of this BRAF inhibitor. In this case, they're looking for, when I knock out this element of a regulatory region, how do I affect the expression of this fetal hemoglobin? And this I actually do in a flow cytometry-based screen. So they do an antibody staining for fetal hemoglobin. They do cell sorting for either things that when you knock out that element, either increase hemoglobin or decrease it, and then they actually call hits based upon which regulatory element modified the expression of that gene. And this is the type of data that they get out of it. So if they take, thousands of guides and they tile them all along these regulatory regions, and they ask for which things actually affect the expression of those hemoglobin, they can pull out a locus like this, where they actually see differential expression actually in human cells compared to mouse cells, where you have the specific region which has a GATA1 transcription factor binding site. So you can actually map important regulatory regions for the transcription of genes by using Cas9 to tile and in principle, create deletions that prevent the binding of those transcription factors. And so in the end, they can, take these sort of data points where they have modifications in expression of a gene, and then relate those to the binding sites that have been determined through ChIP-seq, as Luke described, where you can actually now try to functionally annotate which of these transcription factor binding sites or 3D contact loops are important for controlling gene expression. So this is a significant departure from the typical protein coding screens that have been done. Now you're really asking quite complex questions in some cases about which of these different regulatory control elements drive expression of a gene. Another really nice example comes from John and Max in Jonathan's lab together with Daniel Lim. In this case, so I think Luke had described this briefly in his previous talk, but what they were able to do is to target long non-coding RNAs in cells. And so this is actually a uniquely incredibly powerful application of the CRISPR-I system. So if you think about it, CRISPR-I has been very useful, and I'll show you some slides in a minute where the same screens have been done extremely well with CRISPR-I. But what's nearly impossible to do with an active Cas9 screen is to actually target these non-coding RNAs. So these are in some cases very long RNAs which are not coding for a protein. So you might put an active Cas9 in the middle of it, but what would that actually do to the function of a very long RNA for which particular elements may not be important? In this case, they were able to use CRISPR-I to actually target the promoters of those long non-coding RNAs and screen for essential gene phenotypes. So some of this data I think Luke showed, but the general principle was that they were able to do growth screens in a range of cell types, targeting the expression of those different long non-coding RNAs, and we were actually able to find some very interesting differences, different cell types that had different long non-coding RNAs that were essential for growth. So this is an example of a screen in which they were scanning the protein coding region. So this is to get at the question that was asked earlier. How would you design a guide RNA that was especially bad? In this case, they were actually looking at the sort of the opposite effect. So, how would you design a guide RNA that was especially good for targeting this chromatin modifier, BRD4? So what they did was to take guide RNAs really tiling the entire protein coding gene and look for guides which, when they knock it down, actually cause a growth phenotype in these cells. And the interesting thing that they found was that when you put a guide RNA in some of these really conserved known functional elements of the protein, so these are actually some of the active elements of the protein, these tend to be much more impactful when you knock them out. And the thought is here that in some cases you have a guide RNA that targets the middle of this region, and with a significant probability you actually don't create an indel or a stop codon. Whereas these guide RNAs, even with an in-frame deletion, may have enough of an effect that if you have a very highly conserved kinase domain, for example, you may lose a single amino acid, and that may have an enormous impact on the function of that protein that would not be present if you were to lose a single amino acid here. So their rationale is by targeting highly conserved functional regions of proteins, you can actually have more effective gene deletion. And in some cases, they've actually found this allows you to map the functional domains of a protein. So if you didn't know that these were the important functional domains, you might actually find them by tiling across a gene and actually seeing, well, this is the place where the protein really hates to have an indel placed, or even an in-frame deletion. Okay, so a really important caveat that I think should be kept in mind for a lot of these screens with active Cas9 is that you are putting an active nuclease in the cell, massively overexpressing it, and expressing guide RNA. So this is something that's, in some sense, maybe especially important in the context of a screen where you're constitutively expressing Cas9 protein and a guide RNA over a long period of time. In some therapeutic applications where you're transiently expressing a guide RNA or an RNP, it may not be as important to consider these types of off-targets. But actually, in this case, what a number of groups have found, this is work from Bill Hahn or a group at Novartis. We've actually found quite similar things in our own data, and a number of groups have found this, including Sabatini early on. What they showed was, if you look at the behavior of guide RNAs across a chromosome in a cancer cell line where you have clearly amplified regions, guide RNAs that target these amplified regions are extremely toxic, whether or not there's a gene there. And this is because if you have enough copies of a piece of DNA, you can actually cause a significant amount of DNA damage. In some cases, so significant that the cell will just effectively die because you have this massive DNA damage response. So this is especially important given that many of us are using these highly amplified and messed up cancer cell lines to do our work in. In K562 cells, for example, BCR-ABL locus is massively amplified. It's critical for growth. It turns out if you put a guide RNA in between the amplified genes, you can actually still see massive cell death. So this is something that needs to be kept in mind when you're doing these screens. We've actually found even a single cut has a measurable effect on growth. So I'm showing you a library that we built to try to make an effort at controlling for this effect. So we built our own sort of genome-wide CRISPR cutting library, where we had a set of negative controls that targeted what we thought should be safe regions in the genome. So these are regions that have no annotated, there are no genes there are no chromatin marks there are no regulatory elements, there are no long non-coding RNAs. So across 170 cell lines, there's nothing at all there that should be what we think functional. And we put a bunch of controls there with the idea that we're going to see what the effect of cutting at one site would be in the genome. So this is the work of David Morgans and Michael Weinberg in the lab. To cut to the chase, the sort of interesting observation here is what I'm showing you here is the distribution of guide RNAs for either gene targeting guides. So you can see there's a range of phenotypes. Some of them are very highly deleterious when you knock them out. Some of them are slightly protective. If you compare the phenotype of a non-targeting guide RNA, so this is a guide RNA that we computationally predict should not have any match in the genome with tolerating up to 3 mismatches, versus one of these safe targeting guide RNAs, which should have exactly one cutting site in the genome. You can see that the distribution of those safe guides is actually considerably broader than the non-targeting guides, which means that actually these have much more of an effect than non-targeting. And so you can actually even see this when you look at the substitution position. So depending on the mismatch position, the effect of those safe targeting guides actually differs, which reflects known properties discovered by Jennifer and others about where Cas9 can tolerate a mismatch. So the take-home message is you should actually, if you're doing a Cas9 screen or even an individual retest experiment, if you're going to use a control, use a guide that cuts somewhere in the genome, because there is a measurable effect on growth and some actually measurable DNA damage that occurs. So don't use a non-targeting guide because it's not really an appropriate control.
This effect probably depends a lot on the cell line you're using, because p53 mutants, for example, more sensitivity to single frames.
Yeah. That's definitely right. In fact, you can see that here. So we did it in, I believe, 3 different cell lines, and you can see in some cell lines it didn't quite matter as much. It was not a perfect correlation with p53 status, but you're absolutely right. In some cases, it matters quite a bit more than others. Yeah. Okay. So fortunately, Luke gave a fantastically complete introduction to CRISPR-RNA, so I'm not going to describe this in any more detail than he just did, other than to say those significant problems with toxicity due to nuclease cutting can largely be eliminated by using either CRISPR-I or CRISPR-A to do these screens. So this is, because you're not using an active nuclease, you're using the dead Cas9 just as a targeting domain, you can really, to a pretty large extent, ignore those off-target cutting effects that have been, a significant consideration. In some cases, it matters a lot more than others, but one of the major advantages of this approach is that you don't have to worry about the DNA damage you cause with an active nuclease. And so, as Luke mentioned, there are several different strategies to do this. I'm not going to belabor that point. The other thing that we found, even on our own screens, is that the point that Luke made about the importance of finding the position, the right position to put those guide RNAs, is something that I think everyone can attest to. And so what I would say is if you're interested in doing these CRISPR-I experiments, the new and improved library is really significantly new and improved. It really actually works a lot better than the V1 library. And so I think one of the major improvements was just annotation of the transcription start sites, if I'm correct. So with an improved transcription start site annotation, you were able to do, the significant improvements in the algorithm that Max made, they can now very effectively affect transcriptional repression across the genome. And so these libraries work actually quite well. So here's a couple of examples in 2 different papers, so in this one and the new one. So one of the screens they did early on was one of my favorite toxins, the ricin toxin, near and dear to my heart. Don't need it, but actually, it's really an amazing toxin, actually. So it gets endocytosed, it traffics from the endosome to the Golgi to the ER. It gets unfolded, recognizes a misfolded protein by the ERAD machinery, retrotranscribes translocated back into the cytoplasm where it refolds, and only then can it kill the cell by depurinating ribosomal RNA and shutting down translation. So it's this really kind of amazing toxin that does all these neat trafficking and folding events. And we could use that to find all the genes in a genome-wide screen that were involved in its trafficking or processing. What was shown in this really nice figure is that you can actually see, depending on whether you knock down with CRISPR-I or activate with CRISPR-A, the expression of these previously demonstrated ricin hits, you can see an opposite phenotype, which is really cool because it means that you can not only recapitulate some of the same biology that was found in other screens, but you can actually be even more confident if you could flip the phenotype from repression to activation of sensitivity, for example. You can be even more confident in the phenotype of that gene. And this is just to show, using the same kind of gold standard essential genes, in the new and improved library, you can see a near perfect recapitulation of essential genes with the CRISPR library. So these libraries work really quite well, equivalently well to the best CRISPR active Cas9 libraries that we've used. Okay, so in the last bit, I'm going to describe a number of different sort of applications of these screens. So one really cool application that was just published, so you're going to get a talk, I believe, tomorrow from Tom Norman, who worked together with Britt Adamson and Jonathan's lab, and together with Aviv Regev and a number of other labs, they had described very recently this kind of extraordinarily powerful readout for a screen, which I'm not going to go into in any great depth because I'm sure Tom will do it. But just to highlight, in the context of screens that you might do, it's now possible to not just look at a single gene phenotype, protection and viability, or the activation of a reporter, but to look at effectively the modifications to the transcriptome. So a really high-dimensional multiplex output to go with a multiplex input of a genome scale screen. So Tom will describe that in more detail, but I wanted to just put this in the context of screens because it really, in principle, expands the range of the phenotypes you could look at. So yeah, in the last little bit, I want to describe two applications that we've been pursuing in my lab for these types of screens. So as I mentioned, everyone and their mother is doing some kind of a screen. You end up with a long list of hits, and you might say, well, I know what the first 10 do, but what about the next 50? How do I actually understand at a systematic level how these genes actually work together? And so early on, when I was a postdoc together with Martin Kottman and Jonathan's lab, we had done a genome-wide shRNA screen for ricin modifiers. And what you can see in this map is that I'm displaying the phenotype of a single gene in combination with a series of second genes. And so if it's yellow, that's that's better than expected. So the combination of the two genes when knocked down is a much better phenotype than you would have expected, and blue is significantly worse than you would have expected. And so depending on the nature of that interaction, it can tell you something about how those genes actually work together. So the really cool thing about these maps is if you cluster genes based upon the similarity of this pattern, what you can effectively do is reconstitute a lot of the biology of the cell. So we did a genome-wide ricin screen. We took a lot of the hits from that screen, and we clustered them together in this map by their function. And what you can see is that many of the genes, even if we didn't know what all those different trafficking steps were, we could have found them because here are all the ribosomal genes, here are all the COP1 vesicle trafficking genes. But the really cool thing is if you have a gene of unknown function. So here is C5 or 44. Previously, we had no idea what this gene actually did. But we can see that genetically it looks like a member of this vesicle tethering trap complex. And so one of the really nice things about this was it came up with a prediction for the function of this gene. Genetically, it has a pattern of interactions that looks like another complex that we do know the function for. So now we have a prediction for what this thing does, and we could actually show that indeed this is a physical member of this complex, and in fact defines two new complexes that are subcomplexes of the trap. So these were very powerful tools for kind of a systems level understanding of what the hits from a screen would do. So in my lab, we're interested in applying sort of a new application of this pairwise genetic interaction screen, in this case to look for specifically cancer drug combinations. And so why might you want to do this? Of course, cancer drug resistance is a very widely appreciated problem. There's this famous picture that many of you have probably seen of this unfortunate patient who presented with a melanoma. He was treated and had a really nearly miraculous response to this BRAF inhibitor, which completely shrank all of his tumors. But as almost always the case in cancer, what ended up happening was eventually the patient relapsed and all the same tumors came back and were now resistant to this BRAF inhibitor. So there's an enormous interest in the pharmaceutical industry, and this has of course been an interest for a very long time, in trying to find combinations of targets that would allow you to prevent the escape of these cancer cells to drug resistance. In addition, of course, it's very expensive to develop these kinds of drugs, and so if you could repurpose existing FDA-approved drugs, there would be a lot of utility in that. And of course, testing all those combinations, even if you did know how they would work together, is an enormous effort. And so what we wanted to do was to actually model drug action by using CRISPR to target the targets of FDA-approved drugs. So we wanted to systematically look for drug combinations by targeting pairs of genes for which they had a corresponding target. This is the work of Kyo Han and Edwin Zheng in the lab, very talented postdoc and a great graduate student, who were helped by David and Amy. And the idea is we generate a very large library, in this case 500,000 pairs of guide RNAs that target combinations of genes which have drug targets, and the idea is, can we pull out synthetic lethal combinations that might be effective in cancer? So I will skip over a lot of work that these guys did to express pairs of guide RNAs. There are now a number of different systems that get around some of the problems that Luke described to express pairs of guide RNAs. So we built one, the Weissman lab built one, the Ventura lab built one. I think, as Luke pointed out, it's nice to have some different options for how to do these things, because one of the key problems is if you have identical promoters, you can't actually sequence the guide RNAs directly, and you get a lot of recombination between the guides. So in the end, we built a system that can express pairs of guides that we can actually do deep sequencing on, directly sequence the guide pairs. And what we did was we took genes which had a corresponding drug according to these drug databases. We selected ones that by themselves were not lethal, because if a single gene kills a cell, you're not going to find synergy. And we took genes that had a mild phenotype. We selected the 3 best guide RNAs from our genome-wide screens, and we made a pairwise combination library, in this case targeting about 207 drug targets, which in the end gives you roughly 500,000 pairs corresponding to 20,000 possible drug combinations. In the end, the workflow for this is exactly the same as all of the screens that I've described to you. We now take a double guide library instead of a single guide library, and we infect that into cells, and we calculate interactions based upon how different is the expected pair from each individual gene when you knock it down. And we want to find the really rare synthetic lethal combinations that completely kill a cell only when you have those 2 genes knocked out. So the data are highly reproducible. It doesn't matter if you flip the guide from one position to the other. The replicates are highly reproducible. And here's what the data look like. So largely you have a huge cloud around 0. So what I'm showing you here is the genetic interaction score in one replicate on this axis and another replicate on the other axis. And what you can see is that by and large, most genes actually in the set that we took don't interact at all. So they have 0 interaction. They're exactly what you would have expected. And this is probably not surprising, because we really took a random set of genes that have nothing to do with each other. They just happen to have a drug target, and we decided to look and see which of them, when you knock them out in combination, actually are synergistic, and the answer is very few. What's reassuring is if you knock out a gene and then you use a second guide RNA against that same gene, now the phenotype of the double mutant is actually better than you'd expect. That's because once you knock out that gene, it doesn't matter if you target it with the second gene for the most part. There's effectively the same phenotype, and so it's better than you would have predicted. But what's really nice is we could pull out these rare, actually quite potent synthetic lethal combinations, which when you knock out the 2 genes together, those cells really have a hard time growing. So in the end, we decided to focus on this combination of a BCLX long inhibitor and an MCL1 inhibitor. These are 2 anti-apoptotic genes, and you might say to yourself, well, of course, 2 anti-apoptotic genes are going to be synthetic lethal. In fact, cancer cells differ enormously in the extent to which they depend on particular anti-apoptotic genes for their survival. And so it was actually interesting that we could pull out this pair that happened to also have 2 pretty effective drugs that can be used against them. We also did find a number of other, pairs which were synthetic lethal. Some of these make perfect sense. These are 2 different isoforms of AKT, 2 different DNA damage repair pathways, and so we felt like we were getting legitimately synthetic lethal combinations. So to make a long story short, we were able to show that the combination of these two drugs, corresponding to the two genes that we found to be synthetic lethal in the screen, were actually quite potently synergistic when we used them together. What was really cool, though, is we could actually see that, so here's K562 cells, where we're treating the cells with these two drugs in combination. You can see really potent synergy. When we use these either EBV transformed LCL cells or primary cells, so these are CD34 hematopoietic stem cells. See, the pair that we found were just universal synthetic lethals. They were actually